Hello, everybody. Can you hear me all right in the back? Is that good? Okay. So thanks for inviting me again, actually. It's my second time here on this conference. I think first time was in 2010 when I just had started my lab. So it's good that since then all the lapses that I had during my presentation have been forgotten and you have been inviting me again. So it's a pleasure to talk about our progress that we've made ever since. So I want to talk about engineering molecular systems of molecules using DNA. So the key goal of our lab, our dream, to put it differently, is actually to build artificial molecular machines. Artificial molecular machines that can achieve complex functionalities, much like those of our natural role models like ADP synthesis, for example. Wouldn't it be great if you could build an artificial ADP synthase, something like that, and any other molecular machine that could come to your mind? Or wouldn't it be great if you were able to build smarter drug delivery vehicles that copy some of the capabilities that viruses have, but that would be fully under our control because we have built them, engineered every aspect of that. So I think that would be great, and that's sort of our driving force, this goal. But we have a problem. Or let's put it differently, we have a challenge to solve. Is today, mankind doesn't even know, or doesn't understand how to, doesn't have sufficient skills to build sufficiently sophisticated molecular structures to even realistically think about constructing an artificial ADP synthase. Right? We can study the natural molecular machines, but it's very hard for us to create something that has similar capabilities. So what we're trying to do is we're trying to mimic and adopt nature's principle for making such molecular machines, which is stated here, sequence programmable hierarchical self-assembly with polymers. We're trying to adopt that principle to create such sophisticated molecular structures that one day, hopefully, we can call actual machines. And we're not working with amino acids, actually. We're working with nucleic acids and DNA in particular, because that's a molecule that we understand fairly well. And as you will see, hopefully, there's a chance that actually we can use DNA to actually create machines. So what we do in the lab is we sit on our desks and we design the sequences for lots of DNA single strands so that these DNA single strands, when we throw them together in a single pot mixture, they come together and self-assemble into a designed and a pre-programmed structure like the one shown here, which is a model. Every thick cylinder that is oriented horizontally is supposed to indicate a DNA double helix. And then you can also see these vertical thinner cylinders. These are supposed to indicate covalent phosphate backbone connections. So these are effectively holiday junctions that hold the structure together internally. So this object has no particular function other than to have a particular shape. So we call it a pointer, but you could also call it the middle finger, frankly, if you look at this. But so the whole purpose of this shape is to be completely asymmetric, because this was of advantage when we try to solve the structure of that object. So here is how such objects actually look in the cryo-EM. So you see the cryo-EM density map of this particular object. So we had 82 DNA double helices in our design, and we found 82 DNA double helices. We had 377 of these connections that hold the helices together. We found 376 of them. One got lost. We already learned that our structures are more like rigid porous meshes with lots of tunnels and cavities. So the helices, because they're negatively charged, they try to get away from each other in between the junctions. And then we're going to slice through the structure now, and you will see sort of the cross sections of individual DNA double helices, and you can see how these helices periodically form connections with their neighbors, and this is how the entire object is stabilized internally. So then we are going to make another slice now in a different direction through the object. You can see that for this particular map, the resolution goes up toward the core of the structure, so we can actually see major minor groove of individual DNA double helices. So in this particular case, we could actually fit an pseudo-atomic model into the electron density, which helped us to analyze in detail the geometrical properties of this particular object. So one question, for example, that we had is when we design such an object, how accurate will be the positions of which likelihood will certain DNA base pairs actually show up at certain positions in space. So I've showed you this object mainly to illustrate how these objects actually that will show you as cylinder models look in reality. It's the only high resolution structure that we have at the moment for the entire field. Everything else is basically coarse-grained AFM images or TEM images of individual particles. So whenever I show you a cylinder model, you have to do a mental computation and convert the straight helices into these wiggly, noodly DNA double helices. And also, I mentioned already that this object that I showed here, it has no particular function other than to have a particular shape. So it's a sculpture essentially. But we don't want to be artists, right? We don't want to create sculptures. We want to create machines. So how can we go from machines, from sculptures to machines, to engineering actual machines? So we have a roadmap that we're following, and we're trying to tick off and trying to establish all the basic capabilities that we think are necessary toward actually building machines. So there are several aspects. These are not all aspects that are important here. So you need a method to encode shapes and sequences. I think we have that. We need to be able to make these objects of sufficient quality and yield. I think we have new protocols that can deliver that quality. And then you may want to go to higher order assemblies, more complex assemblies. Such a machine like ADP synthase is a multi-protein complex with different modules, so each module has different functionalities. You must be able to design actual mechanisms with controlled degrees of freedom. And if you want to do catalysis or something, or even molecular recognition, something like that, you need to be able to design with atomic accuracy. And so all of this needs to be done, and ultimately we have to think about ways to actually power these machines. So I really like talking about the progress that we're making along this roadmap. It's a lot of fun. I'm going to show you a few things that we have realized recently. So what you see here is a giant gear self-assembled from a set of 9,000 DNA strands. It integrates about 400,000 DNA base pairs, and the molecular weight is 240 megadalton, so that's 100 times larger than a ribosome, but still a defined size structure, and it's assembled bottom up from sequences. So only thing we did is we designed sequences, and then it self-assembles into this giant object. What you see here is a tilt series of that object as seen in a microscope. So this actually works quite well. We can produce such gears and with relatively high yield. I'm just showing you this that we have the actual perspective to create defined size, rigid, higher order assemblies from a limited set of subunits, so that we can think about, for example, creating artificial capsids and things like that from such DNA building blocks. Or I would really talk about also about the progress that we're making in designing mechanisms, like this one here. We see here is a little you can think of this as a little coffee mill or something like this. It's sort of a precursor for a rotary motor, right? So when you want to make a rotary motor, you first need to be able to make a rotary mechanism. That doesn't have the motor, but you need to have the mechanism in place. So this is a mechanism that has a rotor and a stator, and the rotor can rotate in the stator. Diffusively, there's an energy input. So we made it, and we studied the dynamics of this object in fluorescence microscopy, and what you can see here is the tip of that rotary mechanism sort of tracing out arcs in real time. So it's a rotary mechanism. It does have no directional bias, so it's simply a stochastic random walker, but we have this mechanism now, and now we can start thinking about ways to bias the directionality, couple the movement of this mechanism somehow to, for example, a non-equilibrium chemical reaction. And then we would have a motor. So that's just to see the perspective of this. But today, I don't want to talk about this kind of methods development that we're doing. I want to talk about something else. I want to show you how you can use self-assembled DNA nanostructures to your advantage to perform precision measurements of biomolecular interactions and structure. So I'll do this. I'll report basically on two projects that we have recently completed. One actually just appeared last week. So the story is about a dissection of the stacking forces that occur within DNA double helices. So let me give you some background first. So what you see here is a cartoon representation of two DNA base pairs stacked on top of each other in the context of a longer double helical DNA domain. So what you see here is actual sort of B-form, canonical B-form DNA geometry. So you have the lower DNA base pair. This happens to be a G, C, and then there's the next one, which is an C is an A and E is a T, so stacked on top of each other. So there are two main contributions to the stability of DNA double helices. One bond type, surely everybody learns this in school, is a hydrogen bonds. So you can have 3 hydrogen bonds between GCs, and you can have 2 hydrogen bonds between ATs. But this is not all. Actually, a big part of the stability of DNA double helices comes from stacking forces. So from an attractive interaction that acts along the axis of the helices and sort of in between these bases. So I can ask a simple 2 questions. How strong is such a stacking bond actually? What's the relative contribution of the stacking bond to the DNA double helix stability? And you can also ask a kinetic question. What is the lifetime of such a stacking bond? If you knew that, then you would have some interesting information about the dynamics, internal dynamics of DNA double helices. But we don't know the answer to these questions. So the problem here is that if you look at a DNA double helix, the structures always includes both the hydrogen bonds and the stacking bonds. So how can you study the stacking bonds, stacking forces in isolation? How can you study the interaction between base pairs without actually breaking hydrogen bonds? So here's one way you could do this. You could prepare solutions with blunt-ended double helical DNA domains. And between these blunt-ended DNA blunt ends, stacking forces should occur. So you could think that the system would actually have a bound state in which these 2 terminal base pairs stack on top of each other. And in fact, this actually works. So if you concentrate these duplexes to extremely high concentrations, like 150 millimolar, which is effectively a solid almost, you can shift this equilibrium to the bound state. So yes, there is this attractive interaction, and all know about blunt end DNA ligation. It only works because yes, there is this transient blunt end interaction. So, but 150 millimolar is a really weak interaction. Right? This is really hard to study, and if you want to study kinetics, lifetime of that bond, that's even harder. So we had an idea how we could approach this problem. So the idea was to take multiple blunt-ended DNA double helical domains and put them together onto a solid support, but with control over the orientations of the ends. And now if you could prepare another sort of a binding partner, also having blunt-ended DNA double helices in a defined orientation, also mounted on a solid support, you could think that based on avidity, this system now should have an equilibrium shifted to the right-hand side. And if you understand the stoichiometry in the system, you should be able to conclude something back about the properties of the individual bond. Okay, so this is sort of schematically how this is implemented in a context of such a DNA structure. So we have cantilevered DNA double helices that protrude from a multi-helix bundle, which is shown here just as a cylinder model. So these protruding helices are also coupled laterally, and we control the orientation of the terminal base pairs. So then this beam is coupled by a flexible polymer to another beam on which we have another set of such protruding DNA double helices, again with terminal blunt ends. And the system is designed such that if these things would come together at these interfaces, continuous B-form DNA double helices could form. If any mismatch in orientation would sort of destroy this idea. Okay, so then this this is our reaction chamber, and this is integrated into the context of much longer beams. So the double beam system is actually 500 nanometer long. It is functionalized with 2 different modifications at the other 2 ends, so that we can stick 2 micrometer sized latex beads to both ends of these beams. And then these 2 beads in turn can be held in a dual beam optical laser tweezer, and then we can watch. We can slightly nudge with the laser tweezer on these 2 beads, and we can read out the extension of the double beam system as a function of force. And whenever the system is in a bound state, the extension is shorter. When these bonds break, then it's a bit larger. And this is something that we can track with high resolution in the system. So this is how this looks like in an electron microscope. So these are some of these beam particles with the bonds broken. And here is how this looks like when the bonds are actually formed. So this is, in this case, they look like rigid beams. But when the bonds are broken, it's like a floppy beam. Okay, so let me show you some exemplary single particle data. So this is how this looks like in an optical trap. So we have these two beams. We have these two beads held in the laser traps. We track the positions of the beads, and we're measuring sort of the deflection of the beads out of the trap center. And so whenever the deflection is large, the construct in between is shorter, which would correspond to the bound state, the closed state. And whenever the deflection is smaller, then the construct is larger and it's in the open state. So what we see is that the system fluctuates back and forth between two discrete states. One state, which having the larger deflection, which we assign to the closed state, has a smaller noise amplitude, while the lower state here has a larger noise amplitude. This reflects directly the mechanical properties of these two distinct states. When the stacking bonds are bound, the entire system is like a rigid beam. Brownian motions are suppressed, noise amplitude is smaller. When it's broken, noise amplitude is larger because it's more floppy. So, and then what you can see here is sort of at a particular pretension, we get a situation where the system spends most of the time in an open state, and then occasionally it jumps back to the bound state, dwells there for a while, and so on. If you reduce the pretension a bit, the equilibrium shifts. Now the closed state is visited more frequently. If you go to even lower forces, then it's mostly populated. The system mostly populates the bound state. So this is when this is what you see when you have 4 AT base pairs mounted on the left-hand side, meeting 4 AT base pairs mounted on the right-hand beam. So if you take an array with 6 AT base pairs on the left-hand side, meeting 6 AT base pairs on the right-hand side, the picture shifts drastically. So let me just switch back here. So this was the time scale here, 2 seconds, right? And now we're going to go to 6 of these bonds. Now this is the time scale, so we're looking at much lower kinetics and also higher forces. So this is the impact of avidity. So now what we do, we collected this kind of data for all possible sequence combinations of putting one particular base pair on the left-hand beam and one particular base pair on the right-hand beam, and we collected also different array sizes. And for all of these constructs, we collected this kind of data. We collect the statistics, so of the times, how often the system spends time in a particular states to determine the average lifetime of the bound states as a function of force, okay? Which gives us then this tabulated set of data. I'll briefly explain this. So on the y-axis, we have the average lifetime of a stack array in a closed state as a function of the applied force in the optical trap. This is sort of the range of timescales that we can measure with the optical trap. So we can go from milliseconds to a few seconds. Above seconds, the system is limited by drift. And below milliseconds, we just simply cannot discriminate the states anymore. So then there's a color code here. You can see this. So there's like, if there's a red dot, that means we have, this is a data set obtained with 2 blunt ends. Or orange means 4 blunt ends, blue 6 blunt ends, and green is 8 blunt ends. So you can already see, based on the many colors that you can see here, that there's a big sequence dependency in the stacking bond strength. So here, for example, when you put a terminal CG meeting an AT base pair, actually to measure anything useful, we had to study variance of 6 or 8 stacking bonds in the system. But if you do this with GC on CG, this is actually very stable, and there you can actually measure 2, just 2. This was good. But if you now integrate, if you try to study system at 4, this was beyond our capabilities already. It was too stable. So there's a huge sequence dependency here. Okay, some noteworthy details. So we have here some symmetry in the system. It's called the dyad pairs. So if you look at this sequence combination, so you have a double helical domain on the left-hand side with a terminal AT meeting a double helical domain on the right-hand side with a terminal GC. If you rotate this by 180 degrees, you get this, right? And if you study this, you are studying the exact same molecular surfaces. So you should get the same result, right? So, and in our system, we can track the identity of the left-hand beam and the right-hand beam. So what we did, so what we have in our system, we have variants where we have an AT on the left-hand beam and then GC on the right-hand beam. And now if we place a CG on the left-hand beam and a TA on the right-hand beam, we should get the same thing based on the sequence symmetry. And this is actually what we get, fortunately. So this is, for example, such a diode pair. We get the same behavior. Another diode pair, 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 same behavior. So this gives us confidence that we are measuring the right thing. And yet another diode pair. So then there's another interesting situation. So this is called the swap pairs. If you have here you have an AT on the left-hand side and a CG on the right-hand side. Now if you just swap the sequences, you're basically interchanging the surfaces that were previously buried to exposed, right? So if I have here, the right-hand side of that base pair was formerly exposed, and this construct is buried, right? So now we have different marker surfaces that interact. So in here, you don't necessarily expect that these systems should behave the same way. They could behave the same way, but not necessarily. And in fact, that's what we see. We see both things. We have swap pairs. Here's a swap pair, a CG on an AT. If you now swap the sequence, an AT on a CG, it behaves completely different, saying that the stacking interaction between these surfaces really depends on how you stack them on top of each other. But then this combination, again, is very similar. It's a swap pair. So we have both. They can be very similar and also very different. So I'm just click through this. Discussion of the swap pairs. And come to some further implications. So now that we had this data from controlled arrays of stacking bonds, we use a statistical model to extract kinetic properties from the single stacking bond. This is sort of the result we got there. We plotted here the average lifetime of a stacking bond, or the average dissociation rate for a stacking bond for all these sequence combinations. So this is in a way redundant because it still contains the diode pairs. And these are sort of ranked according to the stability. So here on the left-hand side, you have the variance with the longest lifetime, the most stable ones, or the smallest rate. And here are the variants with the fastest rate. So these go up to 40,000 events per second, actually. Very rapid dissociation. And there's an interesting pattern that comes up if you look at this. So there's some kind of chemical pattern. So for example, the green dots here, so the weakest combinations, this is when you put a pyrimidine on a purine, you get a weak stacking. While if you turn this around and you put a purine on a pyrimidine, you get strong stacking. And the other combinations are somewhat in between. It's kind of a curious pattern. And if you look at this a bit further, in this table, which actually gives you the free energies of stacking, which you can also determine because we have rates for unbinding and restacking, then you see that actually this correlation of strength really seems to have something to do with the atomic overlap. So let's look at this example. So this is the weakest stacking combination that we had in our data set. And this is minus 0.8 kilocal per mole, really weak interaction. So CG on an 18, you can see. So what we're looking along the DNA, the axis of a DNA double helix. So the green base pair sort of is above, right? And then the next base pair underneath is tilted by 34 degrees. So it's like canonical B-form DNA geometry. And if you look at that, you see there's really little atomic overlap between this base pair and the next one. So here's a little bit of overlap. But this one here, the GC and the CG is the strongest stacking. There's a lot of atomic overlap. So there seems to be like a correlation of stacking interaction with the actual atomic overlap. Okay, so this our data set is very useful for us because we understand the kinetics of the stacking bonds, and we can use this to our advantage when we try to design mechanisms. But there might also be some interesting implications for molecular biology. So I like to think of stacking forces as these lightnings. So that's what this picture is supposed to illustrate. So we found the average rate with which these stacking bonds sort of breathe and dissociate is on the order of 15,000 events per second, and is really insensitive to forces along the helical direction. So now, if you think about DNA replication, for example, in E. coli, that happens with a rate of 3,000 base pairs per second. So the replication is actually slower than the rate at which base pair stacks would spontaneously dissociate. So in replication, you need to separate base pairs, which means you need to break hydrogen bonding and stacking. So, and what this data tells us that actually the internal dynamics of DNA double helix might be such that the enzymes involved in replication don't even need to exert forces to actively break base pairs. They could act as a passive Brownian ratchet that just walks along and waits for the next opening. So at least unstacking is not rate limiting. Okay, so then I'd like to come to my second project I'd like to talk about, and maybe in the next 7 minutes I should be able to do this. So this is about uncovering the forces between nucleosomes using a DNA origami gizmo. Some background, what this is about. I mean, you all know that the genome in higher order organisms is highly structured. So at the lowest level hierarchy, you have DNA double helix. Then you have the nucleosomes, and then these are like sort of arranged like beads on a string. And then there is something called the chromatin fiber, which is even already highly debated whether it actually exists in vivo or not, and what the structure of that chromatin would be. And then there might be higher levels of organization which are yet unclear but actively researched. So let's go back to the chromatin fiber. So this is really an ongoing discussion whether or not it actually exists and what shape or structure it might have if it exists. So we thought it could be helpful if we understood better what the actual interactions between two nucleosomes could be and how strong they are. So in particular, so the key questions we're asking in this study is, what is the strength of this interaction between two nucleosomes? How does it, what is the range of that interaction? Does it depend on the relative orientation? And is there also some dependency on the chemical state of the nucleosome, so acetylation, for example? So if you had the answer to these questions, maybe we can make some interesting predictions about chromatin structures. So we're going to use the following experimental approach. We're going to take two nucleosomes and position them relative to each other in 3D. We're going to restrict the relative movement of these nucleosomes to a one-dimensional reaction coordinate. We're going to exert slight destabilizing forces, as you will see. And then we're going to analyze the relative movement that these two nucleosomes do in terminal equilibrium, and that will teach us something about the forces that act between the nucleosomes. So, and to do this, we have built a device in which we can place two nucleosomes that looks like this. We have two rigid multi-helix, two rigid beams consisting of multiple DNA double helices bundled together. So these are these two beams, and they are bound together. They come together at the hinge, which has torsional stiffness. And then we can place two nucleosomes in the system. And the utility of this hopefully will become clear soon. So this is just to show you that this actually works. So you can, this is a variant of this device as seen in a TEM with a pair of nucleosomes mounted here, and they stack. And this is another variant where we put the nucleosomes further away from the hinge, and they also stack. So actually, these images directly demonstrate, yes, there's actually an attractive interaction between nucleosomes, because without them, we don't see that. Okay, so now some more details. How do we get the nucleosomes into these objects? So we created modified nucleosome core particles in which, rather than using a continuous template DNA, we used a branch template DNA from which a DNA single strands protrude, which sort of creates a nucleosome that looks like this. You have these single-stranded handles that protrude from the disks, and this way we can hybridize the nucleosomes to particular positions on our DNA origami support. So now can we somehow quantify these nucleosome interactions? So this is what we did. We took the naked spectrometer, we actually call it a force spectrometer, so the naked force spectrometer, and we collect TM data, single particle TM data, like this one here. So all these particles, we sort them, and we measure the opening angles, which gives us a distribution like this. So this is sort of the frequency at which these particles, without anything in between here, populate particular opening angles. Okay, so now we're going to put in the nucleosomes and repeat the experiment. Again, we collect single particle data. Now with the nucleosomes, we carefully categorize our data that we only look at particles that having two correctly integrated nucleosomes. And again, we're going to measure the opening angles, which gives us this distribution, which now looks different. Right? So there's a new population here coming up. So if you look at the particles, for example, that are located here in the peak, you see that these are those particles where the nucleosomes are very close to each other. Yeah, and this is just another image showing a little zoom up. You can actually see these two coils of DNA around the protein core. So now you can do a different experiment. You can move the nucleosomes further away from the hinge, and then the distribution changes. Here now we get a peak that's located at smaller angles, and this is corresponds to particles that look like this. In order to get these nucleosomes in direct contact, the object has to close further, right? And this actually requires more work, and that's why we see fewer particles adopting this particular state. Which brings me to energy landscapes. So at thermal equilibrium, the probability to populate of a system, to populate a particular microstate, is connected to the energy of that particular microstate. If you have such a distribution, probability density for populating a particular angle, you can just take the logarithm of that, and that will give us the underlying energy landscape, which you see here. So now, if we take the log for the angle distribution that we got with the nucleosomes, then this would be the corresponding energy landscape. And since energies are additive, we can just subtract the contribution from the naked spectrometer and compute the difference, and this will give us a nucleosome pair potential, which is just the energetics of nucleosomes interacting with each other. So this is now plotted in units kilocalorie per mole as a function of the nucleosome center of mass distance, so somewhere in between the center of mass, so sort of the direction coordinate here. And there's a minimum located at the distance where they are in direct contact. And what we see is that this pair potential has a really long range. It goes up to 6 nanometers, which happens to correspond to the contact length of these histone tails. And what we also see that it's really shallow. I mean, it doesn't look shallow here, but 1.4 kilocalorie per mole is really a weak interaction. So you can compare the sort of free energy of a single base pairing event. That's really weak, but it's for the entire nucleosome stacking on another nucleosome. Then we can play games. We can ask how does the orientation matter, the relative orientation. To study that, we made another variant where we rotated one of these nucleosomes 180 degrees, repeated the same experiment. This is the wild type pair potential, and this is then the other pair potential we get with a rotated nucleosome, and looks the same, actually. There's a slight shift here in the minimum. This can be explained by the fact that the clashes actually occur in the other orientation a bit earlier. So based on that experiment, we decided not to study orientations any further because it didn't seem particularly interesting. It doesn't seem to matter a lot. So then instead, we looked at modifications. So this is what we did. We looked at nucleosome variants where we cut off all the histone tails, so called tailless, and we also looked at the variant where we just acetylated a lysine at position 16 in the tail of H4 protein. So what does the acetylation do? It removes a positive charge at this lysine and makes it neutral. So it's just a single charge and that will be changed. Okay, again, the wild type pair potential. And now if we cut away, if you take off the tails, the interaction gets reduced drastically. I would even say this is noise, so then the range is also affected. So it's essentially shut off. It's really weak now. And if you now look at the variant that has the acetylated lysine 16, it almost looks identical to the variant that has no tails whatsoever, which tells us that the bulk of this actual interaction really comes from this one lysine sticking somewhere on the other nucleosome. And you can switch on and off the nucleosome interaction by acetylation. Okay, some conclusions. We have quantified this nucleosome pair potential. It's really weak interaction, 1.4 kilocalorie per mole. It's really long-ranged. And both aspects, I think, make me speculate that chromatin fibers, if they exist at all, are structurally not very well defined. They will be very dynamic due to this squishy potential and the weak interaction. And regarding chromatin structure, so if this is the pair potential, so the attraction between nucleosomes would need to pay for further penalties that you would have in the system. So if you think about structural models that involve DNA bending, I think these are highly unlikely. Okay, then nucleosome orientation doesn't seem to matter. And this H4 tail is really key for the interaction, this has been known previously, but now we see it directly, the impact. And this is regulated by the acetylation of this lysine 16. Okay, so then just to see that you can really play with this, you can also put the nucleosomes in a different orientation, sort of upright into this object, like we did here, and we actually made a stack, sort of like a tire stack. Sort of we have 4 slots in which we can put nucleosomes, like 1 or 4 or 2 in different relative positions. And we're using this at the moment to see whether unwrapping and unstacking sort of are somewhat coupled. It could be that if nucleosomes are stacked, they're easier to unwrap or harder to unwrap, things like that. Okay, so I'm almost done. This is just showing you a field of view image of these particles as seen in a cryo-electron microscope. So this is basically how we collect our data. These are the particles. You have the proteins in there. You do the categorization. And I hope you have seen that these kind of custom-built tools, they really add an interactive dimension to imaging, which I think has some interesting, there are many interesting systems that you could study that way. In fact, I want to use this opportunity to make a little ad. So we have actually we have lots of ideas, interesting projects we would like to work on. We have also a new toy to play with to do actually to get high resolution data. So we have this new Titan Cryos instrument, and this is one of my students. That's not me. So actually it looks the same, but they all look like me. So if you're looking for an exciting journey, we have ideas. We have lots of work to do. We also have the means to do it, but we need people. And with this, I'd like to thank the people who worked on this. So these were the 3 students. Fabian Kilcher has been a PhD student for 6 years in my lab. He did all the stacking work. It was really the Passion Kilcher, as we call it. And it's wonderful, but it was really a great project. I'm happy that it came together in such a big way. And Christian also contributed there, and Jonas Funke was the one working primarily on this spectrometer nucleosome project. Sorry for being late. Thanks for your attention.